procedures, each carries a different fragment. Okay, and after that, I told you we induce into bacteria. Okay, I told you we transfer it to a host, a bacteria, which is our host. Okay, after we introduce into bacteria, we transfer it to the bacteria, we plate out, and after we have 3 different colonies. Each colonies has different parts, different fragments. So we don't want this, for example. We don't want this. Our ideal one is this one. Our desired one is the second one. Okay. So this is somehow wasting our time. Yes, we waste our time and we have problem of selecting the right one. How can we correct this situation? Do you know? How can we correct this situation and clone the best part, our ideal one, our desired fragment? Do you know up to our last session explanation? May I repeat my question? Listen. I told you here we will have 3 different colonies after all these procedures, okay? And we want one of the colonies because this one is our ideal fragment, okay? So we have problem here in selecting the right one, okay? How can we solve this problem? And do not clone these 2 that we don't want them. Do you know? Can you tell us a solution? No. What did I told you about amplification of this specific size? Do you remember, Fatima? What did I tell you about amplification of a DNA? Remember? I told you if we PCR the fragment, we will have the same specific DNA fragment in a huge amount of size. Remember? You don't remember about PCR? We told you. Here it is. We talked about the PCR. You didn't study? I told you. What is PCR? By the PCR, we can amplify a specific DNA fragment. Okay? We can amplify a specific DNA fragment. For example, 30 million fragments from one specific region. You remember now? For example, you want to clone one gene from the specific bacteria. Okay. you should design a primer, forward primer, and reverse primer. Okay, forward primer and reverse primer from that specific region. Look here. And after the amplification, we have almost more than 3 million copy of that gene from one specific copy. Okay, so now here, this is our problem. Look, this is our problem. This is the selection problem. If we cut this DNA fragment of E. coli, look at this, we have different parts of gene. If we cut them, we have too many fragments. You can see them. But which one we want? We want this one. We want triple A. Okay, after its cloning, we will have different plasmid which is cloned. Okay, how can we solve this problem? You can see here, if we PCR our specific, our desired region, for example, we want to clone triple A. How can we solve that problem? We can design specific primers, forward and reverse primers for this specific fragment. Okay, after the PCR, we have millions of these AAA fragments, and then we can clone this fragment. Understood? You all, do you have any question? So please, if any parts you don't understand, please tell me. I can repeat it for you again. Okay? If you have any question in any parts, please tell me. Okay? Fatima, look. I told you, if we cut the region, if we cut the DNA, we have different fragments, but we have one of these fragments. And if we clone them, by the procedures I told you in the previous session, we have different colons. But what do we want? We want one of them. Okay, how can we solve this problem? Look at here. If this is the genome of E. coli, if we cut the DNA of E. coli, we have different fragments. But we want this AAA gene. How can we just have this site of DNA? We can amplify the region by the PCR. We can amplify the region by the PCR. We design a specific forward and reverse primers. We amplify, look at this, we amplify this specific fragment By PCR, we have several millions of this specific fragment. Okay, after that, what do we do? We can clone these several millions of fragments. Do we have any other fragments that we don't want? No. We just have our desired, our specific region fragments. Do you understand, Fatima? Thank you very much. Are the diagram required to memorize or for illustration? Look, the diagram and the illustration is necessary for your understanding, for your better understand the lesson. Okay, it's necessary for you to understand the lesson. So, if you don't have any questions, we can go to chapter 2. Okay. Okay, in this chapter, we want to talk about the vectors. We want to talk about the vectors and gene clonings. I told you about the vectors in the previous session. Yeah, I told you about the vectors. I told you a vector we call hamil, or I don't know, what did you call it in Arabic? Vector? I told you to translate it. Hmm? I'm asking the Arabic translation because of your better understanding, okay? So if you know, please tell me for your from the other students. So we have ناقل. Okay. Yes. Thank you very much. So we have different type of vectors. We have different type of vectors. No, the vectors, yes, the vector is in transmission. The vector transmits, yes, but the vectors is not. Okay. We have different type of vectors. For example, plasmids, I told you in the previous session, and bacteriophage. What else do you know? What else do we use for vectors besides plasmids and bacteriophage? Anybody? Who knows? I told you plasmids and bacteriophage is two types of vectors. Beside them, we have viruses. Yeah, we have different type of virus for the vector. As the vector, we use them as the vectors. So we have three types of vectors: plasmids, bacteriophages, and vectors and viruses. For example, did you have the vaccination for COVID coronavirus? Did you have the vaccination? Who had the AstraZeneca vaccination? Who had AstraZeneca vaccine? None of you? None of you had AstraZeneca vaccine? Which vaccine you used? Do you hear my voice? Okay. So we have class activities for your numbers. Okay. For your grades, we have class activities points. So if you don't be active in the class and if you don't have your seminars, you won't get your grades for the class activity. Okay, so please be active in the class. No matter if you answer wrong, but please be active. I died until I could understand the lecture and could not complete because of time. What the question, please? And so you don't listen to me? I told you. Uh, which vaccine did you use for the coronavirus? No, this is a very common question. Which vaccine did you use? Pfizer? Did you use AstraZeneca? Who had AstraZeneca vaccine? Who had AstraZeneca? No one? Anybody of you, do you know the mechanism of AstraZeneca? Anybody knows the mechanism of AstraZeneca? No? As you know, Pfizer is RNA-based vaccine. Yes, you know it's RNA-based vaccine. Sinopharm is a dead virus. Yes, Zolfaqar, Nahi, you use Sinopharm. Sinopharm is a dead virus, okay? Pfizer is RNA-based. And what about AstraZeneca? Who knows about AstraZeneca? AstraZeneca use a virus as a vector. Use the virus as a vector. How they transfer a corona gene into a virus called adenovirus. Do you know that? Coronavirus, AstraZeneca is cloned. How they transfer a gene of corona into an adenovirus. So what is the adenovirus? Adenovirus is a vector, and they transfer it to your body. Understand? This is the very simple and common example that I can use it for you for the vectors. So if you study about the mechanism of AstraZeneca, you can see we have adenovirus in this vaccine, which we transfer our gene in that. Is it clear? Okay, thank you. So, as I told you, we have different parts of vectors. First of all is plasmid. We want to talk about the plasmid. I told you plasmids are circular molecules of DNA that lead an independent existence in the bacteria. So they replicate um, independently. Here is the plasmids. You can see here is plasmid. This is bacterial chromosome and this is plasmid. بساز شامپانز adenovirus. Yes, Marve. Exactly. So the adenovirus is what here? Is vector. We transfer a coronavirus gene into adenovirus. So adenovirus is what? Is a vector. So as I told you, as you know, we have 3 types of vectors. Okay. Now I'm talking about the first types, which is plasmids. Okay, here it is. You can see plasmid. I told you double strand, circular DNA fragments. We call plasmids. You can see in the picture. And this is the bacteria chromosomes. So I told you they can replicate independently. What does it mean? It means they don't need the bacterial chromosome to be amplified, to be replicated. In the one bacteria, they can replicate independently. So they are circular molecules of DNA that lead an independent existence in the bacterial cell. They carry one or more genes that are responsible for a useful characterization of the bacteria. For example, they can um, uh, they can live in the concentration of antibiotics. It means they can be they can have the resistance for antibiotic. For example, chloramphenicol. For example, ampicillin. So the plasmids may have the antibiotic resistance gene. Okay, how can we use, what is the importance of this resistance gene? It's because we call them select marker, selectable marker. We use the antibiotic resistance as a selectable marker. Did you study this one in your bachelor degree? You remember this one? The antibiotic resistance in vectors, plasmids? Okay. And the other important part of plasmid is they have origin Look, if here is plasmids, they have origin. What does ori do? What does ori do? Tell me please, what does ori do? Origin. Origin is the site where the replication starts. Yes? So, what does it mean if the plasmids have it means that they can replicate independently. Yes? So, they have some specific characterization. For example, they have antibiotic resistance, independently in the bacteria. We have different, so many plasmids in one bacteria. Look at this one. This is a plasmid we call RP4. This is plasmid we call RP4. Look, this RP4 have three different gene of resistance, antibiotic resistance, ampicillin resistance, tetracycline resistance, kanamycin resistance. You can see it. Is it clear? RP4 has three different resistance gene: ampicillin resistance, tetracycline resistance, kanamycin resistance. So, after we transfer it to a bacteria, if we add the antibiotics to the medium, look, if the yellow ones is cell without plasmid and the blue ones are cell with plasmid, after we add the antibiotic, which one can live? Only what? Only cells that have the plasmids. Yes, Fatima, only cells which have RP4. So, look, did you understand? Okay, so we can use it as a selectable marker, for select, transfect, and untransfect bacteria. Return, you want to see this picture? You want me to repeat or not? You understand, Marve? Okay, look at here. I told you plasmids have the specific uh, selection markers. For example, they have antibiotic resistance. So here is the example. RP4 has 3 different antibiotic resistance. For example, ampicillin resistance, tetracycline resistance, kanamycin resistance. So it has 3 different resistance to 3 different antibiotics. After we transfer it to E. coli, some of the bacteria have the plasmids and some don't. Okay, the blue one is the cell with plasmid. The yellow one are the cell with no plasmids. How can we select the bacteria who had the tetracycline? After we add tetracycline, we can collect and we can specifically collect the cells which has the plasmid. Okay, Marve? Here it is. So I told you plasmids have the origin, their specific origin. Okay, their specific origin. What does their specific origin means? I told you the specific origin is for their independently amplification. They can multiply independently. Okay, so what will happen after the cell division? What will happen after the bacteria divide into the plasmids will transfer to each cells. Yes, the plasmids transfer to each cell. But look at here. This is the plasmid and this is the bacterial chromosome. What happened here? What happened here? Look. The plasmid inserts in the bacterial chromosome. Yes, you can see the plasmids insert. No, it inserts here. You can see it in the picture. It inserts in the genome of the bacteria. So the chromosome carrying the integrated plasmids, it goes into the genome of the bacteria. So look at here. We can have 2 different plasmids. This is non-integrative plasmids, which don't enter the bacterial chromosome, which multiply independently. Okay, transfer. And here is integrative plasmids, which integrate in the bacterial chromosomes. Is here. Look, we call them episomes. So, what is episome? Episome is the plasmids which integrated to the chromosome bacteria. And after the cell division, it's amplified by the bacterial genome. Please answer my question. Does the plasmids here amplify independently? Here in the episome type, does the episome or does the plasmids amplify independently? No, because it's integrated in the bacterial genome. Every time that the genome of the bacteria amplify, the plasmid will amplify too. So here they don't amplify independently. Understand? What's what's it benefit? You know, what's it benefit? Episome. It can be stable. Episome can be stable. You know, if we have independent plasmids, they can lost in the cell division. They can be lost. But this type of episome can exist more. The stability is more here. So, the definition of, I don't understand your question. Okay, here in this table, we have some plasmid examples. We have some plasmid examples. Look at here. If we want to tell about some specific characters of the plasmids, we can talk about their size and their copy number. Their size and their copy number. The size of the plasmids are very important. They should be less than 10 kb. Less than 10 kb. The the very good plasmids, okay, should have the genome less than 10 kb. Less than 10 kb. So let's see about this one. Which one is less than 10 kb? Look, this one, this one. So they are short. So the best plasmid size is to be less than 10 kb. Their size is in the range of 1 kb to 20 of 250 kb. As you can see, the largest plasmid is about 250 and the smallest plasmid is about 1 kb. So the best size I told you should be less than 10 kb. What about the copy numbers? What about the copy numbers? As you know, do you you think that we should have more plasmids in one bacteria or less? If we want to clone a gene, if we have, is it better to have more plasmids or less plasmids? Okay, as you know, the copy numbers of plasmids should be more because we have more specific DNA fragments, and after that we have more product. Okay, so the copy number can be from 1 or 2 up to 50 copy number. So, as I told you, if we want the best cloning vector, the more their copy numbers are, the we have the large quantities, and after that we have the more recombinant DNA. After that we have more product. So it's better to have the high copy number for the plasmids. Look at here. This is PUC hashed. PUC hashed is one of the plasmids. One of the plasmids. What is the PUC hashed? PUC hashed is the resistance plasmids. I want to talk about plasmid classification. Okay, now we want to talk about plasmid classification. Plasmid classification. Plasmid classification. The first one is R plasmid. What is R plasmids? R plasmids are resistance plasmids. What does it mean? It means they carry genes which has the antibiotic resistance, one or more antibiotic resistance. For example, the resistance to chloramphenicol, ampicillin, or maybe they can resistance to mercury. So our plasmids are very important in clinical microbiology because they can spread their population and they can use as a treat. I told you RP4, here it is. RP4 is one of the antibiotic resistance, is one of the R plasmids. Which one? PUC hash. I'm sorry, PUC8. PUC8 is one of the R plasmids. So what is R plasmid? R plasmids are resistance plasmid which have one or more antibiotic resistance. Is it clear? The second, if F plasmids F plasmids F F plasmid is about fertility. What does it mean, fertility, in Arabic? Get back fast. Where? I told you R plasmid are the plasmid which contain one or more resistance gene. For example, antibiotic resistance gene, mercury resistance gene. Okay, they are very important role for the bacteria. For example, I told you PUC8 is one of PUC8. I'm sorry, PUC8 is one of the resistance R plasmids. RP4 is one of the resistant plasmids or R plasmids. Is that mean any PCR polymers meet with bacteria? Mean any PCR polymers need? I don't understand. You mean what I told you in the start of the session? I told you about cloning. We amplify our DNA fragments. We amplify or PCR our DNA fragment. Now we don't talk about PCR. We just talk about the plasmids here. Look. Okay. So after that, I told you F plasmid. What does F plasmid means? F plasmid is from fertility. F plasmids have TRA genes. They have TRA gene. Thank you, Iman. Iman is a male name or female name? You know, in Iran, we use Iman as male. Sometimes it's female. Okay. Okay, thank you. So here is male name. I told you F plasmids fertility are the fertility plasmids. They have TRA genes. Look at here. This is the plasmid which call them F plasmids. Plasmid, sorry. F plasmids. They have TRA gene. What will they do? They make pili. You know, of course, the pili in the surface of a bacteria. The single form is pilus in the surface of a bacteria, and after that, they can transfer their DNA to other bacteria. So we call them plasmids, which their DNA can transfer to other bacteria. What's this process mean? What's this process name? Do you know? What do you call this process? Transfer plasmid to another bacteria. We call conjugations. Did you study about conjugation? لا الافتران. Okay, so yes, we call them conjugation. What is conjugation? Conjugation is to transfer the plasmids to another bacteria. This is donor cell and this is the recipient. Thank you. The recipient cell. We call this one F cell or female. We call this male, or we call this cell F positive. We call this cell F negative. So this is the donor cell. Which gene is in the plasmid? I told you, Trojan. Trojan is responsible for transferring the plasmids. So as you know, this conjugation occurs in the bacteria and provide a high variance of diversity, high range of the diversity for the bacteria. Okay. So the second I told you is about fertility. We can have it in E. coli and other is plasmid. Plasmids. Plasmid. What is plasmid? Plasmid. There is a substance called colicins. Colicins is the plasmid. Is proteins which kill the bacteria. Okay, so there is a substance called colicin. Colicin can kill the bacteria. Plasmids are resistant to this colicin. They are resistant to colicin. Which is colicin? Colicin is a substance which is toxic for the bacteria and it can kill the bacteria. What is the example? Here, col E1. Look, the example is col E1. So, I repeat again. Plasmids is the plasmids which are resistant for colicin. What does colicin is? The colicin is a substance which can kill the bacteria. The example is col E1. Another type, degradative plasmids. I will write you here. Degrade. I told you. I'm sorry. I told you incorrect. Degradative plasmids. What does that mean? You know, if there is some toxic substances in their environment, in their medium, for example, toluene or salicylic acid, they can break them. So, if there is some toxic substances, for example, toluene or salicylic acid, what is type? What's the example? Tol is resistance for toluene. Toluene is toxic for the bacteria. So, degradative plasmids can degrade the toxic substances, for example, toluene or salicylic acid. Now, do you have any questions? Okay. Another type, I write it here, virulence plasmids. Virulence plasmid. What do you call in Arabic virulence? Virulence is what cause disease, yeah? What cause disease? Zarova, okay. So, which one is virulence here? Which one is virulence here? This one. PTI ACH5, or it's better to say TI plasmids. TI plasmids. Did you studies about TI plasmids? This is one of examples of TI plasmids. So, virulence plasmids confer pathogenicity on the host bacterium. These include TI plasmids, as I told you, and they can cause disease in the bacteria of Agrobacterium tumefaciens, which induce Crohn gall disease. Crohn gall disease. Crohn gall disease. So they can cause Crohn gall disease in the plants. Which bacteria they live in? They live in bacteria of Agrobacterium tumefaciens. Did you know about Krongal disease? Do you know about this disease in the plants? No? For example, this is a plant, okay? They can. This is tumor. This is tumor. So they can cause tumor in the plants, one kind of tumor. We call them Krongal disease, something like this. Should have studied in your bachelor degrees. I don't know why you don't. Okay. Do you have any questions up to now? Do you have any questions? Do you want to have some rest? Okay, thank you. Just have 10 minutes break and then we'll come back. 10-minute break, okay? Thank you. See you. Are in the eukaryotes, but there is
هل
one
لديك أي أسئلة تريد الحصول
type
على
of
قسط من
plasmids. There is one type of plasmids which is not just in the bacteria. They live in Saccharomyces cerevisiae. Do you know Saccharomyces cerevisiae? There is just one plasmid. We call them 2 micrometer plasmids, which is in Saccharomyces cerevisiae. As you know, Saccharomyces cerevisiae is a kind of eukaryote cells. So if somebody asks you if only plasmids live in the prokaryote cell, you can say we have one plasmid which live in eukaryote cells, which name is 2 macrometre plasmid and live in Saccharomyces cerevisiae. Okay, if you have question, please ask me, Fatima, and then I will continue. Okay. The seminar we ask for us is the specific time is required from us specific subject. I told you the subjects should be about genetic of prokaryotes, okay, and should be in this kind of titles. And you have 10 or 15 minutes to present your seminars, okay? You have 10 or 15 minutes to present your seminars. You can be ready from the next session. As you know, we have holidays, about 2 weeks holidays in Eid al-Nowruz. We have 2 weeks holidays. After that, we have your class, and after that, you can present your seminar. I told you. You know, I can't tell you all of you the specific titles. Okay, you can find your titles about prokaryogenetics and then make presentation, PowerPoint, and then present about 10 or 15 minutes. Okay, be ready for the next session. Every session we have 1 or 2 seminars. Okay? Thank you. So, if you don't have any question, I will continue to talk about bacteriophage. Bacteriophage. As you know, bacteriophages are the viruses which infect bacteria. Did you study about them? Yes, of course you have grades on it. As I told you, we have class activity grades. If you be active now, here, and answer my questions, and be active in the class, I assign some points. After that, for your presentation, I'll assign some grades for that too. So we have class activity grades. You can choose a topic from the book, and also you can choose a topic from articles. Okay, let's talk about bacteriophages. As you know, bacteriophages are the viruses that specifically infect bacteria. So, like all viruses, phages are very simple in structure. They have DNA, or maybe sometimes they have RNA molecules carrying number of genes, including some replication of gene replication genes and capsid genes. So, as you can see in the picture, we have capsid surround the DNA. Here is DNA. You can see DNA, and it's surrounded by capsids. Will the application be in English? Because we are not good at English. Of course it should be in English. Otherwise, I can't understand. Okay. As you know, students, listen to me. Your course, and you're studying science. Okay, you're studying science. Science language is in English. So it's better to improve your English, because if you want to study PhD or if you want to research, you should read articles, you should write articles. Which language you should read or write articles? In English. So science language is in English. So you're studying science. It's better to improve your English. Okay, Mr. Haidar? Thank you very much. So, I told you this is the bacteriophage. We have DNA, which contains replication genes and capsid gene. It's surrounded with capsids. Of course, if you do your best, you will succeed. These two kinds of phages. Look at this. Did you study about lambda phage? Lambda, yeah. This is phage lambda. Its structure is head and tail. Head and tail. Look at this. We have head here, and here is tail. So we have two different phages here. Here is lambda. Lambda phage has one part, head part, which contains DNA, and we have tail part. What does tail part do? Tail part helps the phage to attach to a bacteria and insert the genome to the bacteria. So, if here is a bacteria, it sits on the bacteria and inserts the genome in the bacteria. Okay, here is M13. M13, look, M13 phage. It's filamentous. It's filamentous. Look here, we have DNA molecules. We have DNA molecules, and it's surrounded with protein molecules called capsids. So, M13 structure is filamentous. Look, this is its picture. And a lambda phage is head and tail structure. Okay, this is two common phages. You can see here, this is lambda. This is lambda phage. You can see lambda, as you can see in the picture here, attach to the bacteria and inject the DNA into the bacteria. So, attach the bacteria from where? From its tail part. Attach to the bacteria and inject the DNA to the bacteria. The second step is the DNA phage. DNA of the phage replicates. So here, the phage DNA molecule is replicated. What happened next? After that, capsid components are synthesized new phage particles, and after that they will assemble. So at first the phage attached to the bacteria, inject the DNA is replicated. After that, capsid particle synthesize, and after that they assemble the whole particle of phage, and then cell will lyse and the particle come out from the bacteria. So they will lyse the bacteria, they will break the bacteria and come out from the bacteria. What's this procedure mean? Do you know? What do we call it? Here, when they lyse the cell and come out. Do you know? Did you study about lytic phase? Lytic phase. Did you study about it? No? Are you kidding me? Lytic and lysogenic. No? None of them? I think you forgot about this too. Of course you should. You must study them in your bachelor degrees. Who knows about these two? Lytic and lysogenic. None of you? Okay. So, I'll explain about it. The phage infected cycle, as I told you, it has 3 steps. The first step is phage particle attached to outside of the bacteria and inject the DNA. The second step is the phage DNA molecule is replicated. After that, the third step is the protein and capsid particles synthesize, and after it will be assembled, the phage particles assemble and release from the bacteria. So all bacteriophage have 2 cycles, lytic cycles and lysogenic cycle. What is lytic cycle? Lytic cycle is when the phage infect the bacteria very rapidly and kill the bacteria and lyses the bacteria and come out like this. So this type of rapid infection is called lytic cycle, as release of new phage particle is associated with lysis of the bacteria. So they will synthesize too many of phage particles and lyses the cell and come out. This is lytic. And what about lysogenic? Look, here is about lysogenic mechanism. In contrast to lytic cycle, lysogenic infection is characterization by insertion of the phage into a genome of a bacteria. Look at the picture. Look at the picture. The phage attach to a bacteria and inject the genome to the bacteria. After that, the lambda DNA molecule integrated in the host chromosome, integrated in the host bacteria DNA. So after that, does it kill the bacteria? No. So lysogeny